introduce Richard Lonsdale from the University of Oxford. His talk title is Synaptic Potentiation Required for Associative Learning in the Adult Fruit Fly?
Thank you. Good morning. I'm going to talk about how flies learn, including some unpublished experimental data from my colleague Pedro Jacob and the model we built based upon that data. And I promise that within the next 5 minutes, you'll have the answer to the question that's up on the screen. If you expose flies to an odor together with some electric shocks, and then you present a second control odor, and finish by giving the flies a choice between those two odors, most flies will avoid the odor associated with electric shocks. And by counting the flies, we quantify this behavior as a performance index, the fraction of flies choosing one odor minus the fraction choosing another odor. We know that this learning takes place in an area called the mushroom body, where Kenyan cells respond to signals from upstream projection neurons. And the mushroom body also receives reward and punishment signals, and its output influences the fly's behavior. We're going to focus on an area called the gamma lobe, which is responsible for expressing memory within the first hour or so after learning. In this highly simplified representation, for each odor, I show just one Kenyan cell to represent all the Kenyan cells that respond to that odor. And you can see 4 output neurons. 2 of these drive approach behavior, and 2 drive avoidance behavior. And when a Kenyan cell is activated, it excites all of these output neurons. Now, for each output neuron, there is a corresponding dopaminergic neuron which modulates the synapses between the Kenyan cells and that output neuron. And the gamma 1 and the gamma 2 dopaminergic neurons, which I'm going to call DANs from now on because it's much shorter, they respond to punishment, and the gamma 4 and the gamma 5 DANs respond to reward. We ignore gamma 3 because that doesn't seem to play a role in electric shock learning. And this compartmentalized structure enables the fly to associate odors with good and bad experiences. So, if a Kenyan cell is activated by an odor, and then we deliver an electric shock, which activates the gamma 1 and gamma 2 DANs, that combination results in depression of the synapses in the gamma 1 and gamma 2 compartments. So this is not Hebbian learning. It's synaptic depression, and it's triggered by excess dopamine. And we can measure this effect. If we perform calcium imaging of the output neuron dendrites while presenting an odor and see the response, we note that after that electric shock experience, the response to the blue odor is much weaker than the response to the red odor, and the blue odor is the one associated with the electric shocks. So what I've just given you is a sort of a textbook story for how flies learn using synaptic depression. But is there a role for synaptic potentiation? I'm going to answer that question by asking a different question. This is the aversive trained learning protocol that we've already seen. What happens if we try to confuse the flies by repeating that aversive learning, but with the odors reversed? And what we see is that the flies now have an aversive memory for the red odor rather than the blue odor, but it's not as strong as the original memory, the original aversive memory for the blue odor. My colleague Pedro has performed calcium imaging of the gamma 1 and gamma 2 output neuron dendrites before and after that reversal learning phase. And what we see is that after the initial aversive learning, as we expect, we've got a weakened response to the blue odor. But after the reversal learning, we've now got a stronger response to the blue odor and a weaker response to the red odor. So here is direct evidence that synaptic potentiation is playing a role in the learning. It's strengthening the response to the blue odor. So, to help us understand and more widely apply these observations, we developed a model which I'm going to describe to you now. It's based upon the circuit we've already seen, and we have a firing rate representation for each neuron. And the dopaminergic neurons, the DANs, they do not excite or inhibit the other neurons. They only modulate the synapses between the Kenyan cells and the output neurons. And to begin with, we include a plasticity rule, which is that if the DANs fire at a high rate, then for any Kenyan cells that were recently active, we will depress those synapses. That's our plasticity rule. So that's the only time that alpha is non-zero. So let's apply this model to the reversal learning that we've already seen. What we find is that the first set of electric shocks, we get synaptic depression, it weakens the synapses associated with the blue odor. And then the second set of electric shocks weakens the synapses associated with the red odor. And if we make the assumption that the fluorescent response to an odor when we did the calcium imaging is proportional to the synaptic weight, we can include that experimental data in this picture. And sure enough, it's relatively easy for the model with synaptic depression to explain what happens in the initial phase of learning, where we get a depressed response to the blue odor. But what the model cannot do is reproduce what happens in the second phase, the reversal learning, where the response to the blue odor is strengthened. So we need to include synaptic potentiation in our model. So the hypothetical plasticity rule we apply to do that is to say whenever there's a high down firing rate, the synapses for all recently activated Kenyan cells are depressed, but the synapses for all other Kenyan cells are strengthened. So now we have a bidirectional plasticity rule. And let's apply that to the reversal learning. Well, that enables us to quite easily have a model that reproduces both phases of the learning. We can get the initial depression for the blue odor, and then we get the strengthening of the response to the blue odor and the depression for the response to the red odor. Great. Now, if we include a third odor in this, just out of interest, a green odor that wasn't associated with any electric shocks, you'll notice that that is now the most attractive odor for the flies. So what might be happening here is that the synaptic depression creates the odor-specific memory, while at the same time, widespread synaptic potentiation creates room for future learning. So even a fly that leads an action-packed life will never get into a situation where it can't learn anymore because all its synapses have been depressed down to zero. Okay, we'd very much like our model to also predict performance index that characterizes behavior. Now, when we present an odor, we can calculate a sort of net approach indicator for that odor by adding together the firing rates for the approach output neurons and subtracting the firing rates for the avoidance output neurons. And if we do that for two different odors and then look at the difference between them, that differential firing rate, D, is an indicator of the fly's preference between those two odors. And we can convert D to a performance index just by assuming this simple relationship here. So in this formula, we only have one parameter, D0, that we have to specify on the basis of experimental data, and we've got a model for converting the output neuron firing rate into performance index. Before I finish describing the model, there's one other thing we need to look at, one other learning situation that's very important. If you subject flies to a series of shocks and then present them with an odor, you find that afterwards they're attracted to that odor. It's almost as if they associate the odor with relief from the end of the electric shocks. But our current model cannot predict that. Our current model doesn't predict any odor-specific learning from that scenario. So we need a new mechanism. Now, it's been observed that when the gamma-1 and gamma-2 DANs are firing above their base rate, at that time, the gamma-4 and gamma-5 DANs seem to be inhibited and are firing below their base rate. So we make the assumption that there is a subsequent rebound for the gamma-4 and gamma-5 DANs when the shocks have finished, and they will fire for a certain period above their base rate. And if during that limited period an odor comes along and activates the Kenyan cells, we have the combination required of high down firing rate and activated Kenyan cells to give us synaptic depression. But this time, it's in the gamma-4 and gamma-5 compartments on the right. So this has reduced the blue odor drive to the avoidance output neurons. In other words, the fly is now more likely to approach the blue odor, which is what we need. So we include this relief memory mechanism in our model. Okay, so the main components of the model are now in place. The key thing we need to do is the various parameters. Every model has quite a few parameters where you're not sure what value to set. We need to choose values to those parameters such that we get a good fit with experimental data. So this is how we've done that. For the bidirectional plasticity model, we use calcium imaging data, and we've got two sets of that. Pedro has got measurements that he took in both the gamma-1 and the gamma-2 compartments for our reversal learning. But we also make use of a recently published paper from the Scripps Institute where they provided some great data from the gamma-2 compartment for various scenarios. The rest of the model is calibrated using performance index or behavioral data. And here again, from our lab, Pedro's got some performance index data for the three main types of learning protocols I've been through. But we also use a paper from Genelia where there is relief memory data. That's Yoshieso and Jerry Rubin. That's already published. Okay, the model is finished. We've got all the parameters set. To finish, we need to test the model on a few new scenarios. So the first one we want to look at is actually that same Yoshieso and Jerry Rubin paper, but we're using data from that paper that wasn't actually in our calibration. And what they did was, rather than deliver electric shocks, they used optogenetic stimulation of the DAN in the gamma-1 compartment, and they presented this at various different times relative to the odor presentation. And what their data demonstrates is that when you present an odor, you have a 30-second window within which excess dopamine will create an aversive memory. And then beyond the 30 seconds, you no longer get that aversive memory. And what's really pleasing is that our plasticity model, which remember was calibrated using calcium imaging data from two completely different set of experiments, is actually showing the same time window that we see in this data from Genelia. Now, the second set of experiments is perhaps more challenging because of the diversity of these ordeals devised by my colleague Pedro, and that's more challenging for the model as well as for the flies. Some of these scenarios involve a third green odor, but the final test at the end is always between the blue and the red odor. And again, we find that relatively, the model does relatively well at predicting the pattern of behavior that we get in the experiments. So what can we conclude from all of this? Well, first of all, both synaptic potentiation and depression are needed. And that combination enables the fly to prioritize multiple odor associations. And if you combine that bidirectional plasticity model with a relief memory mechanism, you can actually explain a fair variety of behavioral outcomes. But I do want to emphasize that the relief memory mechanism we're suggesting is speculative. We don't have any direct evidence that that's actually what's happening. I'd like to finish by thanking my colleagues at Oxford for their support, and thank you for listening.
There's time for questions.
Good morning, thank you. Would you say something, a little more details about the alpha function in the learning that?
Yeah, sure. Yeah, great question, thank you. So the way the alpha function is set up is rather like that data from Genelia, that when the Kenyan cells are activated, we keep track of a trace of that activation, because a Kenyan cell is activated, really it's only the first couple of seconds of odor onset where it has a high firing rate. And we keep track of that initial firing rate, and we combine that with the high dopamine from the DAN appearing, and when you get the combination of the two together, you get synaptic depression. So that's how that alpha function is calculated.
And there's also a kind of an Hebbian part of it, or?
No, it's not Hebbian, because the plasticity, the synaptic depression seems to be independent of whether the output neuron is firing or not. It's the conjunction of excess dopamine from the DAN and the presynaptic Kenyan cell firing rate that produces the plasticity. Yeah.
I had a quick question. Maybe we can set up the next speaker in the meantime.
Yeah, sure.
Does this require, it seems the balance between the potentiation and depression would require that there's a certain number of total synapses. Does it, for stability, ultimate stability, does this sort of require a homeostasis on top of that?
Yeah, I mean, that is a good question, which we just haven't looked at really, but there's clearly a sort of pseudo-homeostatic thing that seems to be going on.
Great. Let's thank the speaker for a wonderful talk.